Good afternoon, everybody. My name is Liesbet Geris. I'm from the universities of Leuven and Liège in Belgium. My background is in mechanical engineering. I actually wanted to biomedical engineering, but that degree didn't exist at the time when I was studying, so I graduated in mechatronics and product design. Afterwards, I did then my PhD in biomechanics, and during my postdoc, I shifted towards my current topic of research, which is tissue engineering. So the talk that I will be giving you today has actually one red thread through it, and that is bone tissue engineering. Everything you will be seeing has to do with bone tissue engineering. There's maybe some other things that are connected, but this is the big connector. And why am I here today? Maybe it's because this is a view on system regenerative medicine. There are some specificities to regenerative medicine, which is that in contradiction to medicine in general, this has a large in vitro part to it. In general, in vitro tools are not necessarily very interesting for the in vivo work, but in regenerative medicine, we aim to build something in the lab that will then be implanted in vivo. So all the processes in vitro are actually very relevant and also need to be studied because we need to control those processes. Not because they might be relevant or have a specific link with the in vivo, but simply because we are making a product that needs to be controlled. Just to situate myself and our group a little further, so I'm attached to the universities of Liège and Leuven. In Liège, we mainly do the computational work. It's a French-speaking university. Leuven is a Dutch-speaking university, and there we do most of the tissue, so the experimental tissue engineering work. There is where we have the lab, wet lab. And then you see Brussels is really very close by, where we hope to get most of the funding or try to get most of the funding from. When I say we, then I talk about all these people. This is Prometheus, so this is our tissue engineering group. I think about 10 of those people are computational or modelers, actually. So my own group, so the people that I supervise, they all are member of Prometheus as well. So and the other members are then mostly either biomedical scientists or MDs or people that work on process engineering and chemical engineering. We are organized like this, so this is actually the overview or the organization of our lab. Every one of these little symbols is like a small group of people working on that specific aspect. So we have people working on the ground substances, which is the growth factors, the cells, and the materials. And then we have people working on the mechanisms of action. So when you put them together and you do something with that, either you implant it, so what is this, the influence of the in vivo microenvironment, how does the tissue form in vivo and how does it interact with the existing tissue? But also how does bone actually develop in an in vivo context? So developmental studies is also part of that. And then we go more towards the clinical translation with process engineering, the preclinical team and the clinical work. And you see this funnel shape. It actually indicates that what we're doing here on the ground substances is way, way wider than what we will ever put into the clinic. So we are not restricting ourselves here to only those things which at this point are certain to make or to be able to make a transition to the clinic. We go wider, and then as we go along, it will narrow down simply because of all the regulatory and other requirements. Here at the bottom you see a number of enabling technologies like imaging, modeling and data management that we're working on to actually support research in all of these different little teams. To come back to tissue engineering, well, this is basically the problem that we're working on. What you see here in green and in brown are the amount of donors and transplants over time, and this is for the US only. And in gray you see the demand for organs, people on the waiting list. And typically when you think about organ transplantation, people think about liver and heart and lungs, but also bone is actually part of these organs that need to be transplanted. Now, bone in itself, usually, actually I'll start with the bottom thing, 5 to 10% of all the fractures of all the defects heal by themselves. When you just do minimal, you have a cast or something like that, but other than that, you don't need to do anything. So bone is a very intelligent material. It will constantly adapt because it has different types of cells that can act as sensors, that can sense the environment, and that will steer reparative processes. Both when you're just like us now standing, there's a constant turnover of your bone tissue. I think your whole skeleton is about 10 years old if you think about it. But there's also, when there is a fracture, there's actually a very active cascade of repair processes that come into play. And this is quite robust. And this is also scarless. So normally after a couple of years, when you look back on an X-ray, you should not find back the location of the fracture that you had in your bone. But as I said, in 5 to 10% of the cases, this does not heal. And then we're talking about people that either elderly people that have problems with mechanics, but you also have people with congenital defects. And this is our patient population that we're working with. These are children with neurofibromatosis. Neurofibromatosis type 1 is a very broad disease, or with a lot of or syndrome, and part of it, and so part of these patients also have problems with their skeletal system. And then usually either in the lower arm or in the lower leg, there is actually a spot where it's really weak bone that they have. And these children will they will be born with a broken leg, or they will break their leg just when they're trying to stand up when they're a few months old, or when they're a year old, let's say. So without any big force, they will break that leg, and it doesn't heal. So what it says in Dutch here, she's walking around for 16 months with a broken leg. This girl broke her leg when she was 6 months old, and finally she had surgery when she was 4.5, because, well, simply with these very small children, you cannot do any surgery in between. So for 4 years, she ran around with a broken leg. And currently the treatments, a lot of them involve, for instance, growth factors like BMP2 that have to be added. Well, recently in January, FDA put a ban on the use of BMP2 in children, except in extreme cases, but there's a lot of side effects, and also it's not very predictive. Other treatment strategies involve placing an Alizarov cage around the leg and doing bone transport, which is very cumbersome, very painful, and a very lengthy procedure for these children. So if we could come up with something that we can implant, like a living piece of tissue that will lead to bone formation, that would be of considerable help there. Now, this is actually what tissue engineering aims to do, creating a living implant. So not an implant that is steel or plastic, but something that is alive, that will integrate into the body, and that will assist or actually replace a damaged or an absent organ and do its job. Typically, you see that kind of image when we talk about tissue engineering, and that means that you take cells from the patient, that is step 1, you put them in culture, step 2, you add a material and growth factors, so chemicals to it, that is step 3. You culture it, in this case very simply in a petri dish in step 4, and then you implant it back into the patient. So these are combination products of cells, carriers, and growth factors. That is the typical thing. Now, what we've tried for a very long time is to combine, well, to look at what you wanted to make, bone. So you took a material that looked a lot like the native structure of bone. You took cells that were residing in the bone, and then you took a growth factor that had to do with bone formation, and you combined them. Now, this was how it generally went. This field was started like in the late 80s. Does anybody have any idea how many products, tissue engineering products, or regenerative medicine products are approved for the European market at this point in time? A guess. Sorry? No. No, it's 5, but I still think that's really low. I mean, for something that has been active for so long, I think it's really low. And if you look at it, it's either cell therapy, so that is only cells, no matrix, or gene therapy, there's no matrix, no cells at all. There is a combined ATMP, which is also mostly cells, a little bit of matrix, and then there are 2 more cells, cell therapy issues. So there is actually no ATMP that at this point combines and a growth factor and a cell and a matrix, simply because it's really difficult to get this or to prove it to the regulators that everything is safe, that you understand the product. So there's a big problem there, that we do not understand what it is that we're making. Actually, when you look at the term tissue engineering, if you take the parallel with automotive engineering, chemical engineering, the first thing you do before you start with the engineering part is look at the first part of the term, and you define what you mean with chemical or automotive or marine or whatever. But we never did this with tissue. We never said what it is that we were going to make. We're just trying something that looks a bit like the tissue that we think is missing. So there's a problem there. And at the same time, we also never looked at how nature actually does these things. We just looked at the end point. So now more and more people start to realize that there's something wrong with the approach that has been followed in tissue engineering for so long, and are going towards a more biomimetic approach. Now, these are classical examples of biomimetic approaches. Now, if you go towards bone, then, well, your biomimetic approach means going at and look at development, how things have happened during development. And for bone formation, essentially you have two main processes, which is intramembranous ossification, which means for the flat bones like in the skull, you have direct bone formation. So you have some substrate, which can be fibrous-like tissue, and then you have cells that directly differentiate into bone-forming cells on that substrate. And then you have endochondral ossification, and you hear the chondro, so the cartilage part in it as well. That is where you first make a cartilage template that then gets transferred into bone. And that is how most of your long bones, so your arms, your legs, and other long bones are actually developing. So those are two examples that we can study to try and optimize our processes. Because that is one last thing. Bone or any other tissue, it's a process. It's not an end stage thing. It is the result of a series of events. And if you look at how it happens in vivo, well, from utero to birth to skeletal maturity, it takes years. And we're trying to cram this into a couple of weeks in the lab, so this is already a challenge. But to try and completely take out the element of time and just say, okay, we take one scaffold, one cell, and one growth factor, mix it together and plant it, that is maybe a step too far. So we should, and we are at this moment trying to go through, for instance, this endochondral pathway. If we make a piece of cartilage in the lab, and we prime it correctly, when we implant it, it might form bone, and try to recapitulate this endochondral ossification. It has some additional advantages because cartilage as a tissue, or chondrocytes, they're much more robust for hypoxia, for instance. So all these questions of your cells that you implant dying off in the first few days before you have vascularization, they're mediated if you go through the endochondral pathway. So, engineering. We want to engineer tissue. So far, in tissue engineering, most of the engineering has been done for looking at very fancy materials and building of bioreactors. But actual engineering of that's about it in terms of engineering tools that have been used in that field. So we are now trying to use models to because that is one essential other engineering tool, to look at how we can quantify and optimize our product or process, and maybe look at what the effect of the in vivo environment would be upon implantation of that product. Now, you've seen the image of all the collaborators, so we're quite a big group and we have experimentalists in our group. So we try to do this, in an integrative way, always combining people that do experiments with people that do modeling. Either first we have the modelers and then we try to set up experiments, or first we have experiments and then we try to add modelers to that group. Whereas there's always a feedback loop. Now, the way I'm going to present our work, I could use different classifications. So one very typical classification is the multi-scale thing. You have models at very small time and length scales, up to models that go to and look at organs. Another classification could be, if you look at the vertical axis, going from phenomenological, so starting from the data, to more hypothesis-driven models. That's also one that we could use. Then another one is going through like a model life cycle, where you have models that maybe look more at establishing a good model, or other questions that need already more work on parameter optimization or sensitivity analysis or predicting new aspects. By the way, this is, if I can be so shameless, this is a figure from a book that David and I have edited on uncertainty in biology. So it should be appearing next year or early next year, late this year. Might be interesting. But the approach that I'm going for today is this one. The 4 C's, so cells, carriers, cultures, clinic. I've distributed the models, or I've grouped the work that we've done in these 4 different headings. And I will go through all of them and explain you actually what it is that we've done, what the questions were, what the model system was that we've used, and how we try to make a contribution. So it's not going to go in depth. We don't pretend to be anything else than applied scientists. So we are taking what is available in terms of simulation tools and model descriptions, and we just apply it to the questions we have at hand. So we're not developing any new ways of doing modeling or looking at data. We're just trying to integrate the things that we think we need in order to answer the question that we have. So starting with the cells. Well, some of the questions, I think everybody is familiar with Waddington's landscape. So a cell is sitting somewhere on the top of the mountain, and depending on the cues that it feels, be it chemical, mechanical, electrical, or otherwise, the cell is going to follow a path downhill somewhere. And now, what is really important is that we understand when we have a cell population and we want to implant it, that we are sure that the cell is somewhere in a stable valley, that the cell is not sitting here between cartilage and bone, and we think that it is bone. Well, effectively, if you implant it, it will go down this side of the slope and it will form stable cartilage. So we need to make sure that it is in a stable state. And especially going from in vitro to in vivo in tissue engineering, that has quite frequently been the problem, that it's very hard to predict where your cell is going based on what you are measuring in an in vitro experiment. So some of the questions are, where is my cell in this landscape? Is it in a stable or unstable state? And then how can we push or keep cells into a specific state? Because these pushing and keeping are 2 different things, in fact. And so how can we adapt our culture strategies for these cells? This slide is here for the benefit of when I present for an experimental oriented crowd that is not necessarily convinced of the value of modeling, simply to show that this is like half of one figure that represents metabolism or part of the metabolism, just to show that these things defy the human intelligence, that you cannot gather this in your head all alone. But I don't need to convince you of that. This is the figure that we have. This is the graph that we work with. The genes and the growth factors and transcription factors that you see here are all related to the chondrogenic differentiation state. What you see in blue are the main growth factors that are known to influence chondrogenic differentiation of mesenchymal stem cells. The green ones are the important transcription factors, and yellow is everything else. The red connections are inhibitions, and the black ones are activations. This is the result of literature search. So there is going to be bias based on what my PhD student who found this has read, and what was available, what had been researched a lot in the literature. That being said, we wanted to see if with this graph we could already recapture some of the essential things in chondrogenic differentiation that we knew from experiments. As I said before, you can keep this at a very phenomenological level, so from a graph all the way to a mechanistic model, and I will be discussing Boolean, additive, and a little bit of ODE work that we've done in this context. So starting with the Boolean, this is a simpler version of the graph I just showed. This was our first attempt that we did. And then the first thing we checked was whether in a Boolean it was a multi-level, so we could be either 0, 1, or 2, whether this formalism would allow to capture actually the stable states that we have in chondrogenic differentiation. And I should have put this graph here. What you see in a growth plate, you see that the cell is going through the cascade of chondrogenic differentiation from a resting state, which is not really well defined, to a proliferative state where it actively proliferates. and then to an hypertrophic state where it basically enters an apoptosis or a suicide program at the end and is preparing the environment for bone formation. Now, this was a Boolean network. It does not have any any temporal resolution, and also the values, well, you're limited to on or off, basically. You can do multi-level, but okay, we wanted to do a little bit more, so we made an additive model, which means that we allowed all the genes or all the factors in there to have a value which is continuous between 0 and 1. And we added a distinction between fast and slow processes. So you have fast processes like the phosphorylation, and then you have the slow processes which have to do with the mRNA. So that is how we made this distinction. So every time we have an update of the network, we have a certain state of the slow processes, and then we look for an equilibrium of all the fast processes. So all the proteins are allowed to update themselves, and then when we have a stable state for the fast processes, then we make another step of the slow processes. That is how we make it's kind of a pseudo time that we can introduce that way. Now, what did it allow us to do? And we tested this not only for this chondrogenic network, but also for a network that had to do with helper cells, T helper cells. We could actually reach stable states that were not reachable by a pure Boolean formalism. So simply having this more fine-grained time aspect allowed us to reach a number of states which were reported in biology, but which are not reachable by pure Boolean formalisms. And another thing you can do is then a dose effect. If you, for instance, increase the dose of a certain growth factor, so you can increase the importance that this one arrow has downstream in the network, then you see that you change the behavior. And what I mean with the behavior is what you see in these boxes are, for instance, for BMP, when it's present, you have it's a proliferative phenotype, so you're in the SOX9 phenotype. When you increase the dose, and it's not very visible, you see that the color of the green is becoming a bit more intense, so you actually increase the basin of attraction of the SOX9 phenotype, of the proliferative phenotype, and then you get this shift. Part of the network, if you do, let's say, 100,000 simulations, part of the network will end up in a zero state, which is neither proliferative nor hypertrophic, and part of it will still stay in the SOX9 state. So you see that you get different behaviors. That is because of the dosing. Now, basically, in terms of analyzing this, we looked at canalization and perturbation. So we analyzed basically if I initialize this 100,000 time with just random initialization, where does my model end up? What are in what stable state is the final state of my model ending up? And then when I perturb it, how does it shift from one stable state to the other? How will we be using that? Well, if here what you see is either a full upregulation or a downregulation of each of the individual components of the model. And green means an increase of the basin of attraction of this Runix state or of the SOX state, so or of the non-state. Those are basically the three states we always come across. Either it's proliferative or hypertrophic or none of the two. And what you see, for instance, is if you upregulate, let me pick, for instance, FGFR3, if you upregulate it, you see that the basin or the chance that your cell will end up in a SOX9 phenotype decreases, whereas this chance that you end up in a Runix2 phenotype increases. Now, if you downregulate FGFR3, then nothing really happens. So you can add it to your culture medium in order to push the cell towards hypertrophy, but once your cell is in a certain state and you remove it from the culture medium, then it doesn't seem to have the same effect. So you see the asymmetry between pushing cells into a state and keeping them in a certain state. We can turn this into a Markov chain that will then depict what are the probabilities of going from one state to the other state. And when we were doing that, we looked at now what are the main little parts of the network that were actually responsible for a number of these transitions. And for instance, one of these was this complex that is BMP driven. So from BMP, you go through a SMAT complex, and then you see this mutual inhibition between DLX5 and MSX2. And actually, when we look back at the data that we used to base the network on, this arrow here, this was derived not from literature on osteochondral differentiation, but from, I think, cancer literature somewhere. So there was no direct evidence that this arrow was, this thing was actually going on in endochondral or in chondrogenic differentiation. So we did the very small tests where we stimulated our cells, just mesenchymal stem cells that we use in the lab, with Wnt, BMP, or a combination of both. And we saw that indeed there was a direct effect of BMP on the presence of MEF2C. So we could confirm that this arrow potentially was correct to have it there. So it's actually this part. So how do you validate this? Because, well, we tried to do a number of normal measures on networks, but we didn't get very far. Doing a whole chipsec analysis on our own was also not going to happen in the short time that we had, or the student had, to finish his PhD. So we did two things. Essentially, we did an ensemble approach. We investigated the relative importance of all the interactions. So all the arrows, now we took them, in order to reduce the parameters, we took them all the same. But what happens if you play with that? So if you have a number of networks where you change the weight of all the different interactions just randomly. For that, we used a genetic algorithm where we just sampled. We said, okay, this is the profile that we want. And then we looked at which combination of weights for all these different arrows will give us actually a good result. In the end, we ended up with, out of the 10,000 analysis we did, with 300 possible interaction sets, which gave us, based on this topology, the good behavior. And then if you look at what these arrows are, how well they correspond to, when you do overexpression and knockout and stuff, to the results that we had with the network that we just derived with as little parameters as possible, we saw a pretty good match. So there is some of these results are different, indicating that we might have to go back and look at some of these results again, because some arrows might not be needed, or they might be indirect and still give a good effect. That was one part. The other part is inference. So we did go into the literature, and we took from a number of these databases, I think 60-something microarray data sets that we had, which were all related to Liesbet. So developing growth plates, either the cells or the growth plate entirely. They might not have been diverse enough to get to be really good as a starting point for inference, but that is what we had to work with. And we did, since we didn't have any preference for any of the inference methods, Johan just went through a number of them, and he tried all of them. And instead of choosing one, we went for the consensus network. So while what he came up with is probably not the best possible inferred network, but maybe the least wrong network. And that we then compared to the literature-derived network that I just presented earlier, where we looked at the true positives, so edges that are both in our literature-derived network and in the inferred network, and then false positives where we had the edges that were inferred from the data but not present in the literature. And then you see that there is actually, well, this is a positive result in the sense that our inferred network goes in the same direction as our literature-derived network. It's not a random situation, which would have been if we would have had the same amount of false positive and true positive errors. What I need to remark here is that we use our literature-derived network as a gold standard, which is not entirely true because that was our choice. So it's kind of it's more a comparison between the two than an absolute validation of this inferred network. These are also very preliminary attempts, so this is work in progress, but this is how we try to come up with some measure of validity for the network that we have. in parts of the lab, people are not looking at the entire picture, but in more specific single pathways of part of the pathways that I just showed you. And especially the Wnt and BMP pathways are things that people look at more in depth. So we also made a small model to encompass more details of those pathways and also more kinetic information. Based on models that were available in literature and some of the data that we had, we made a model of the BMP and the Wnt pathway, and we coupled it in two different ways. So there was here a direct feedback from the phosphorylation steps into the deactivation of the shevet, and then from the GSK complex there's an influence on the last phosphorylation step of the SMAD complex. This is just typical ODEs, everybody here will notice. So you have the rate of change of one of these factors, which is then dependent on formation, dissociation, and destruction, basically. Now, we didn't get very far with this yet. What we did is we corroborated it primarily with experiments from the literature. So what we see in literature, or what has been published, is when you give a BMP pulse, then you see that there is this succession of the phosphorylation steps that you see on the right hand side, and that we can also see these are then the different phosphorylation steps that you see appearing over time. And if you look at the stimulation, the same little experiment I just talked about with the Wnt and the BMP, you see that if you look at beta-catenin, which is one of the factors that are important to then regulate further cell differentiation, you see that there is a mutual inhibition between Wnt and BMP, which is also present in the experiments in the literature. So these are things that we are repeating for our own cells because these are cell dependent. So if we want to do something with this for our own cell type, we need to repeat them. And Mohammed has a poster here that will talk more about the ways we're trying to come up with decent parameter values for these models, as obviously we don't have all the data for all of these parameters. So these are some of the ways we look at our cells and the culture in the lab. The next part that is important are the carriers. What you see here are calcium phosphate based carriers. They're often used in tissue engineering because they are very similar of bone tissue. And some of the questions with these degradable carriers, obviously what happens to this carrier in vivo and how is the biology influenced by the degradation products of these carriers, and how can we match better the cells with the carriers that we're using. We started in this topic with first a screening experiment. We bought a number of these clinically used calcium phosphate carriers. Turns out that data sheets from suppliers are not so very reliable, so we did all the biomaterial characterizations again, mainly using nano-CT actually to look at the structural elements, and then some basic chemistry to look at the composition. We combined them with the cells that we use in the lab, the HPDCs, which are mesenchymal stem cells. We implanted them on the back of nude mice because it was human cells. And then after some time, we took them out and we reanalyzed them. Either we looked at, for early time points post-implantation, we looked at gene or protein data. And then for later time points, we mainly looked at structural, so really how much bone was being formed. And just to show you how we do this quantification with the nano-CT, if you use contrast-enhanced nano-CT, you can really nicely see the distinction between the cartilage and the bone that you get in developing mice. So here we see the histology, and here you see the... don't like using this. And here you see the same image, but this is nano-CT data basically. And the colors are made to match, of course. But we use this now as a routine tool in the lab to do virtual histology rather than wait for all the different histological steps. So back to the experiment. Initially we had 5 different carriers that were clinically used. We had pre-implantation data. 3 days and 12 days after implantation, we took them out to look at the gene and protein data, and 8 weeks we let them 8 weeks implantation period was then used to evaluate bone formation. Then we did some basic statistics to look at which of these data sets actually contained most information with respect to the 8-week result. And it actually turned out that if you looked at day 3, where you combined the material information, so pre-implantation information, with the biology that you can measure at day 3 after implantation, that gave the best prediction of the amount of bone that was formed 8 weeks after implantation. Now we took another sample, so we bought other scaffolds that we tested, and we were able to more or less repeat the prediction, or we were in line with what was predicted, except for this one, ripper bone. But that was, well, for me that was okay, because all these scaffolds were calcium phosphate collagen combination scaffolds, whereas ripper bone was the only one that did not contain any collagen. So basically it had the message, which is important for my experimental collaborators, that experimental data can only get you so far. It can be interesting, but for the data set in which you actually took the data from. As soon as you go above or beyond that data set, you might have to redo your experiment or rethink your analysis. Because these type of models, they can tell you what is going on, but they don't answer the question, why is this going on, or how does it work? So we combined this, or we are combining this now with another type of model, where we try to go a bit more mechanistic. And this is a combination of the level set, or this is a partial differential equation system where we look at the dissolution of the calcium and then the response of the biology on this local calcium concentrations. For that we used a level set description to simulate the degradation of the grains. So what you see here is actually a slice of this calcium phosphate collagen scaffolds. In white you have the grains, the calcium phosphate grains, and what you will see in color is the dissolution or diffusion from the calcium from the scaffold, and at the same time you'll see that there is degradation of the grains. So here you see that there is degradation ongoing, and at the same time there is dissolution of your calcium. This is not something we can currently measure or there are no tools to measure this local calcium concentration. So most of the analysis tools we have to look at the presence of certain factors are actually contain a decalcification step. So that doesn't help us any further with this. So using this type of models is the only way to get a good grip on the local calcium concentrations that the cells, which are seeded onto the grains, are actually feeling. How we can validate is through in vitro dissolution experiments, for instance, which look at the overall dissolution of the calcium from the scaffold for different types of scaffolds. And once we have established then the local calcium concentrations, we can combine this with a more mechanistic model of bone formation that is driven by calcium ions, as you see here. This is quite a simple model where we start from stem cells that can then differentiate into osteoblasts, which form bone, or which form first the osteoid, which is the non-mineralized bone, and then the mineralized version of the tissue, which is more reminiscent of bone. Cells can proliferate, cells can produce growth factors, and that was modeled as a system of partial and related differential equations in FreeFEM. The analysis here are still ongoing because the exact forms of all these, the differentiation and the proliferation still need to be determined. We are doing in vitro experiments to get more data to find the appropriate forms. But in the first set of experiments, or in silico experiments that we did, we found the following. And what you see here in red is bone that is being formed either right next to the grains or in the open spaces between the grains, or just completely around the grains. The reason why I'm putting this here is because these are exactly the 3 types of bone formation that we also find in the lab. So we get some scaffolds, they have bone formation onto the grains, some scaffolds in the open spaces, and other scaffolds in the area surrounding the scaffold. So in that sense, there must be something that we can capture with the model. Now the idea then, to go back to the question, is to design the optimal scaffold for a specific cell type. For certain patients, we might not have a big number of stem cells to start with, and then it is important that you choose this carrier exactly right for that patient or for that amount of cells that you have. What you see here is a graph. On the horizontal axis you have the scaffold, different types of scaffold that you have characterized by the release rate of the calcium from that scaffold. In the vertical axis, the amount of bone formation that you see. The three different colors are representing the amount of cells that you have, stem cells for the patient that you have to work with, that you can see on the scaffold, starting in black from very little cells to orange, the amount of cells that you basically would desire to have, so a high amount of cells. If you look at the black graph, what it tells you is that if you have little cells to start off with, it is important to choose your scaffold exactly right, because otherwise your cells will not do anything. Whereas if you have a lot of cells, it really doesn't matter very much what scaffold you pick, but there is some optimization that you could do by choosing it more appropriately. So in that sense, we have validation of this scaffold already. The only thing is that we have some problems finding scaffolds that are in the medium release rate. So we need to build them ourselves in order to really validate this graph. So the third C is the culture. Culture essentially means that we take our combination of cells and scaffolds and we don't implant them immediately, but we culture them first in the lab. For various reasons, we might want to do that. And the bioreactors that we use are either hollow fiber bioreactors, but mostly, if we use the scaffolds, is a perfusion system. Perfusion system means that you actually have a pump that is pumping fluid throughout the scaffold, from the top to the bottom of the scaffold. In these bioreactors, we have a number of sensors, so things that we can measure like pH, oxygen, lactate, the pressure drop over the scaffold. So these things we can measure. And in chemical engineering, people are coming up with control strategies. So based on these readouts, to control the process of differentiation in order to make sure that what you get after 3 weeks, for instance, is always the same. That you are very precisely controlling this. Now, the problem is that you cannot really know, based on these general readouts, what is actually going on inside your scaffold. This is some kind of homogenized readout, and it doesn't tell you exactly what is going on inside. You can only also guess what the influence of fluid flow is, for instance, on this whole process. Yeah, this is just a picture of a bioreactor. And just to show you what a construct then looks like, by the way, the reason we're now suddenly using titanium scaffolds is simply because they don't degrade over time. If we are studying a process, then it's nice to know that some components at least are staying the same, that we don't need to deal with them right now. Once we feel that we can control this process, we can replace the titanium with a calcium phosphate and add another bunch of variables to the process. But right now we're focusing on how can we measure what is going on, and so we don't want to deal with the variable biomaterial right now. So we have a titanium scaffold, and you see that there is some tissue forming on the inside. On the right-hand side, you can see analyses that look at soft tissue and mineralized tissues, and here you see live-death assays, or also another hexabricks, which is another contrast agent, which also tells you what soft tissues are in there. If you add mineralization to the medium, you can get things like these, which from far look a bit like calcium phosphate based biomaterials. So in order to model the process in the bioreactor, we went in little steps. First, we started with the static conditions. So you have the cells, you put them on the scaffold, and you simulate the growth in a static condition. Now, what was quite remarkable is that whenever you have a corner, cells prefer to sit there and cells grow there. We were not the first ones to see that, obviously. There is this curvature-based growth hypothesis that has been out there for quite some time. So we decided to use that as the basis for the simulation of neo-tissue growth in the bioreactor. So essentially you calculate the local curvature, and based on that you have a speed with which the interface between the new tissue part, the green part, and the void part is moving. So the speed with which your growth factor, your new tissue is actually filling up the chamber. You can do this. We did this in the level set methodology again, and then you can actually simulate quite complex geometries. Up till now in the literature, people have used image-based techniques, which had quite some limitations when it came to complex geometries. But we can easily simulate quite complex geometries, and even when these new tissues come together and meet, then everything is still calculated correctly. That was validated with different types of scaffolds. So we use 3D printing for to make our titanium scaffolds. So we could make titanium scaffolds with different unit cells. So we had hexagonal, square, and triagonal. And also we could play with the pore size of the pores that we had in our scaffold. We used a single calibration point to calibrate our simulations. And then we looked at the results. And they were actually quite satisfactory for the comparison between simulations and experiments. Now, that is static. Obviously, that doesn't do, because if you keep your construct in static for too long, the inside, well, it starts dying off. So we needed to add fluid. We needed to add a CFD analysis. And this slide actually is a very stupid example of how making a model can already teach or change some things in the lab, because for consistency purposes, people had always put, if you have the bone chamber here, it's rectangular. They always put the scaffold at the bottom. Well, it's easy because it's repeatable and you're sure that you always do the same thing. But we also always saw that there was less growth at the bottom. Now, probably we should have thought before or already much sooner that there might be an effect of the inflow. But seeing the picture like that actually made them realize that they needed to place the scaffold, or that they could calculate what the critical height was that you needed to place the scaffold in order to have a fully developed flow, or as fully developed as it can get in that bone chamber, in order to have more consistent results. So that was one change of just doing the CFD analysis. Didn't really mean much, but it changed the way of working in the lab. So then we needed to combine the CFD with the growth of the neo tissue, and we went for a Brinkmann approach, where we could combine Darcy and Stokes. So flow goes not only through the hollow space where there's no tissue, but it actually also goes through the neo tissue, because it's a porous tissue, where it is then simulated by using the Darcy equation. And inside the hollow space, it is a Stokes, because the speed of flow is so low, we can use Stokes. And then we can calculate actually what the velocity is, the flow velocity inside the scaffold. And what you see here, it requires some explanation. In pink, you see the interface between the neo tissue and the void space. So you see that the pink part is actually, we'll start here, filling up the entire scaffold. So that means that this is now completely filled with neo tissue. So everywhere where you see pink, this is the interface between the neo tissue. And what you see is, if you look at the flow rate, in the beginning, when the free flow space is getting narrower, then your speed is increasing because it's a small diameter and it wants to go through this free flow space. But then at the end, it is forced to go through the neo tissue, and then the speed is way lower. So once you have this speed of flow, using a number of assumptions on the permeability and the porosity, the measure of porosity of your tissue that you can get from SEM analysis or something like that, you can calculate shear stresses. In bioreactor studies, if you look at the amount of PhDs that have been done on wall shear stress for the design of bioreactors, it's huge. But what we actually saw that is the wall shear stress in red is not nearly as high as the shear stress inside the neo tissue to which the cells are exposed. But that is something that wasn't really calculated a lot. But now we can calculate this. We know that this will affect the flow, the speed of growth of the neo tissue. So we added not only the local curvature, but then the shear stress. We added that as a factor into the determining of the growth speed, and also the oxygen tension. We know that cells need oxygen, So we added that to the growth rate of the new tissue, where you have this general graph that is depicting the influence of shear stress, where there is an optimum, and then when it's too much, there is no more growth. And for oxygen, well, you just have to make sure that there is a certain amount of oxygen present. We simulated this for the whole bioreactor, and I'm just showing a couple of the analyses that we did. Is, for instance, this influence of the placement of the scaffold inside the bone chamber. If you place it at the bottom, we saw exactly the same thing as we see in the experiments, namely that there is very little growth at the bottom, whereas if you place it higher up, then you see a nice filling up of your scaffold. If you play with the flow rate, when you have a very low flow rate, it means that your oxygen is actually being consumed while the fluid is still inside the scaffold. And so when you reach the end, there is little oxygen and also little stimulus by means of the shear. So you're on the left-hand side of the slope of the shear stimulus. Whereas if you take a bit higher, both the oxygen and the shear stress are way more optimal for tissue growth. Now, recently we added two more things, which is the glucose and the lactate, not because we like to add stuff and make the model more complex, but because these two things are used really a lot in control modules of bioreactor simulations. And what you can see is if you look at specific scaffolds design, in this case it's a gyroid, and the results are, well, the analysis is still ongoing, you see that you get these little pockets where you have very little flow, and where your oxygen is dropping, your lactate is increasing. So you can actually say something about the distribution of the tissues inside your scaffold and how healthy your cells are at the different locations inside your scaffold. Whereas you can calculate, for instance, the cumulative lactate production, which is also what you can measure. So you can relate the overall measurement back to the local situation of the cells. Just to make the point that when people measure, for instance, a pressure drop over the scaffold, they think they can relate this then to the filling of the scaffold. And what you see here, if you look at the pressure drop, by day 10 you will see that it starts increasing. But if you relate this to the neo tissue volume over cell culture, and the bottom line is actually 100% filling, then you see that by day 10 there's almost like 80-90% filling of the scaffold. So this pressure drop is not nearly sensitive enough to actually have a good idea of what is going on inside your scaffold. So that is why a model as an in-between step might give you more information. Okay, that was the third C. Onto the clinic, and these are still very much research-driven questions. So it's not that we're using patient data to do systems medicine. These are still questions from the research side, looking at causes of adversity of fracture healing and possible treatment strategies. Now, this fracture healing model dates back to my own PhD, where we started making a model that had partial differential equation model, working with cells, the tissues, the growth factors that were relevant for fracture healing, and then gradually adding more details onto it. So this is like the first overview of the model, where you see the four central cell types for fracture healing. And again, you have the proliferation, differentiation, matrix production, and growth factor production. Along the way, there were a number of pilots that we used to validate these models. Well, they can all do normal fracture healing, but that is not so very interesting. It's more interesting to see what happens if you do adverse causes of fracture. And one of the first trials that we did is the atrophic non-unions, for which we had a pilot experiment ready, where we actually had, you take the fracture, you remove part of the marrow and the periosteum, which is the skin around the bone, and then there's no healing. There's no stem cells, there's no healing, but there is vascularization. So the question is, at what time, or can you, by injecting stem cells, at some point regenerate the healing process? So we played around with different scenarios. We correctly simulated the non-union after 16 weeks, and then we added stem cells at different locations inside the callus. And we saw that when you add it to the center, well, you get good healing, whereas if you add it onto the sides, you get this cortical shell formation on one side and nothing more. When we compared this to experiments, we actually saw that our predictions were way more optimistic than the experiments. That's on the right-hand side, the two most right columns. You see that for the experiments, there is a large chunk still of cartilage and fibrous tissue present after 16 weeks. And if we look back at the data, then we saw that this cartilage and fibrous tissue was sometimes on the anterior side, sometimes on the posterior side. So if it would have been always on the same side, then it might have been that mechanics was playing a role, but this was not the case. So it didn't seem to be, I cannot exclude it, but it didn't seem to be really caused by the mechanics. So very stupidly, it might have been the way we injected these cells, because we just blindly injected the cells into the callus. And if you inject we checked this with the simulation, if we injected it to the side, well, we also got unicortical healing. So it might be that that is what took place. But this is also just a way in which we used the model. So we got a better correspondence, but still there was something off with our rate of chondrogenic differentiation. Now, we can make this a bit more complex. In the previous model, we had some degree of vascularization as a homogeneous variable or as a continuous variable, but that didn't really mean much. Our experimental collaborators weren't really too happy with that representation. So we added a discrete way of simulating the blood vessels with an intracellular module that was determining where the tip cell, so the first cell of the blood vessel, was going. What you see here are simulations where you only see the active blood vessels. So active blood vessels is when one blood vessel meets another blood vessel, there's a loop and blood can flow, then we have an active vessel. And that's why you see it in little chunks, because it takes a while for a blood vessel to meet another one and then be showing up on the screen. The biggest thing that we're interested right now in the lab are these large defects. So if you have a normal defect, you get good healing. For rats, this is 35 days, for mice 28. But if you look at the right-hand side, these large defects, they don't heal over time. And also in terms of bone formation, you see good healing in 35 days for the normal size fracture. But then for the large defect, we see that there is bone formation on the ends of the bone and then nothing, which is also what we see in the experiments. If you really have a critical size defect, you see capping of the bone ends, and then the biology stops. So basically what is happening here is that our blood vessels are not fast enough. They need to come from the sides, because when you have this large defect, the blood vessels from the muscles are also damaged, and there is no really ingrowth coming from there. And when they have to come from the ends of the fracture, it takes them way too long to reach the center of the fracture. And so the oxygen level there in the center is really low. And even though you have chondrocytes there, which can stand this low oxygen way better, it's still too hypoxic for them, so they check out. And then by the time the blood vessels at some point have advanced further and oxygen can diffuse in, there are no cells there anymore. And then, since there are no cells, there's no growth factor production that will attract the blood vessels, and the whole thing just falls flat. Now, if you would have this contribution of the blood vessels from the sides, you see that it is way easier to get this full bridging of the fracture than when you would have the active blood vessels. So when we start injecting them, again, we did the simple test, and what you see, each data point here is an injection, and then just letting the simulation run. So it's not because you have the same line that we injected at day 0 and then 7, 14. No, these are all independent simulations. Where you injected day 7 and you see what the result is, you injected day 14, you see what the result is. So these can be much refined, but that is the approach we took here. And you see that basically everything you do up until 5 weeks is aggravating the situation. The problem is your blood vessels aren't growing in fast enough, and so if you add more cells, they consume more oxygen, and your whole thing will be dead even sooner. If you add then after what blood vessels are sufficiently developed, if you add stuff, you see that you get more bone formation, but in no case we actually did get good clinical union, which means that the ends actually touched each other. Now, these are just stem cells that we modeled, but you can prime your cells for hypoxia. So you don't need to add stem cells. You can also add cells that have been primed for hypoxia and which have changed their oxygen uptake. And then you see that you do reach, even though it's not 100%, you do see that there is union of the two sides of the fracture. So you need to give the system long enough to get the blood vessel structure in place, and then you need to add cells that can still deal with a relative amount of hypoxia. You can do that, prime the cells, or you can put in place a plug, which is not really injecting, but it is adding some kind of hydrogel, which contains microparticles that are oxygen releasing, and that is what we're building right now. We have the oxygen particle or the oxygen producing particles, and we're going to quantify them, characterize them in the lab, because then they will provide, for at least for the first period, sufficient oxygen for the cells to stay alive inside the fracture callus. And we use the fracture healing model to see what is like the minimum amount of oxygen that we would need and the minimum amount of particles that we would need that create the oxygen to sustain the fracture healing process and carry it through the first difficult period. Now that is one example. We didn't add yet, for instance, like the calcium phosphate carrier together with the cells. What we did do is look at admission of growth factors in different constellations. We looked at the influence of the mechanics that I haven't talked about today. And then, well, we get this, when you have too much mechanical stimulation, there's a non-union. And then when you stabilize it under certain conditions, then you get a recapitulation of the fracture healing again. Now, to come back to my starting point, to the case of the NF1, as I said, it's a quite wide syndrome. So you have people with all kinds of, they all have a mutation in one specific gene, but it can be different mutations, and they have different symptoms. Also in terms of for the people that have skeletal problems, it's not all the same. Some are worse and others are better. But if we look at some of the in vitro data and the clinical data that is available, the problems mainly reside in the differentiation of the stem cells towards the chondrocytes and the osteoblasts, and then everything that has to do with the fibroblasts that are way more aggressive than in a normal situation. So because we didn't know anything about these factors, we decided to go for a design of experiment approach where we did 200 simulations, where we took specific combinations of these 8 parameters that we found to be affected by the NF1 case. So we use a very big word, we had 200 digital fracture healing situations. And the way we represent them here is by using a complication index, which incorporates that when it's really high, when the CI is high, that means that it is a non-union, but it also has a high fibrous component, which is very specific for the NF1 fracture healing case. And what you see in the graphs, each dot is the result of a simulation characterized by only the complication index at a specific time point. And what you see horizontally is the specific parameters. So what you see here are 2 parameters, the endochondral ossification and the cartilage formation parameters, which can vary. And of course, this should be here not 0.5, but 0.05, this one. Which can vary in the range that we have in the model. And what you see is that for a number of parameters, like the endochondral ossification, if you look at early time points, and this is a time scale for mice, so 21 days should give normal healing, that you still have quite a lot of non-unions. So up there, that is non-union. Down here, this is union. And for parameters that have to do with the cartilage, if they go really low, then there is no union whatsoever. For all other values, you see that you can have both union and non-union, which is kind of reflecting the fact that you have this very heterogeneous population. If you wait longer, 35 days, 49 days, you see that some of these points, they will actually come down. So these points have actually moved down, and you get delayed unions for a number of patients. Now, what you see is that you get compensation. And the reason that you have both low and high, so unions and non-unions for similar parameters, is that in some cases you get compensation. So if you have very high or if you have very low cartilage formation, you're in the non-union. But you can also have very low proliferations, for instance, what was I going to say? You can have very high fibrous tissue formation and still be in the union case. It all depends on the specific combinations that you have. So the degree to which this process was affected by the genetic defect. Now, this doesn't teach us much about the clinical case other than that you can have a variety. If you play with these parameters, you get a wide variety. But we're now simulating treatment strategies involving BMP2 addition for these patients. Because if you look at the literature, you will have reports saying BMP2 works great, and then you have reports that say BMP2 doesn't work at all. So there is a big variation, and we hope to get that also from the simulations. But then, of course, as I said, this is really a research tool. The question remains what we can do with this for our patients. Okay, that was the last part. I have a few concluding slides and remarks. One is that I believe that the way we do the models in our lab has helped us to answer some of these questions, or at least have a different view on a number of the questions, and open sometimes the mind of people for other strategies. We can also use it to play around a little bit with the different strategies, see what might be interesting or not. And especially for tissue engineering, it is going to be really important in terms of quality control, because if we want to increase the number of products that we put onto the market, we need to show that we're in much better control of the process. So we need to be able to steer it way better, and we need to know more about it. And as in anything related to modeling, the 3 R's and the personalization might be a plus. Now, this is something that hasn't, or it has been touched upon very briefly. Is this utopia? Well, FDA is already putting in place these guidelines for if you want to include this in your preclinical dossier. And I think the ASME is working on further elaborating this draft. So if you come to the regulator with a preclinical dossier which contains models, then they now have this list of guidelines to make sure that your model is of sufficient quality. So if you have a CFD model, they need to know a certain amount of things. If you have another type of model, they need to know a certain type of things. So this means that it's actually becoming much more real. And also in the States, it has already been alluded that there was an FDA approval for the work of Dennis Noble. Another example is this glucose sensing and insulin delivery device that also works with the model to do the control of the delivery of the glucose. And there is the scientific community. It's not just the Casym, it's also another very similar organization, which is the VPH. They share the same roots. It's the systems biology, Dennis Noble. It's all the same people that were at the beginning. So there are a lot of communities that are working on this to try and make this real. And for me, when I read this, that for me meant that it's really getting real, because insurance companies are seeing how they can adapt their policies and their plans based on wearable devices and models that go with it. So that means that there is actually money becoming involved, and that we're getting closer, I think, to getting something that is actually real. So that for me was the overview that I wanted to share with you. I would like to thank all these people, well, all the people of Prometheus, but especially the students that did all of the work that I showed to you today, and take the opportunity to shamelessly advertise the book that we have edited. So thank you very much for your time.